Hey guys, so Stella and I will be presenting Endo for you this year. And so we decided to do it in a series of short videos. So I'll be doing the intro and hypothalamic, anterior and posterior pituitary hormones today. Then Stella will take you through thyroid, I'll go through adrenal, and Stella will take you through pancreas. And so in each section, we're going to do a bit about the anatomy, physiology, and any conditions that go with that gland. So first up, we'll talk about endocrine signaling. So you've already done a lot on nerves and they have very quick response times. However, endocrine signaling happens a lot slower. So it can take minutes to hours for any actual change to be seen. So endocrine signaling is basically hormones going through the bloodstream to their target gland, which activates more hormones to go through another gland, which activates more hormones to go to the target cells. So they tend to have a low concentration of hormones in the plasma, but there are high affinity receptors in the target tissues. And in certain cases, you can have a neurohormone system. So like for your catecholamines, so your adrenaline and noradrenaline, you have nerves which go to the adrenal glands, which stimulate the secretion of adrenaline and noradrenaline, which then go to their target cells through the bloodstream. So the endocrine system is composed of a lot of parts. So I'd say the central one is your hypothalamus, then your pituitary, so your anterior and posterior parts, because that's where most of the feedback loops start. And then your other main ones that we talk about are your thyroid, your adrenal, your pancreas, and your gonads, so your ovaries and testes. We have three types of hormones. We have peptide and protein, amine, and steroid. So your amines tend to be your catecholamines, dopamine, and thyroid. Your steroids include like your adrenal and sex steroids. So your peptide and protein and amine hormones, they're all produced via DNA and protein synthesis, and then they're stored in granules and then secreted via exocytosis whenever they're needed. Steroids, on the other hand, they're not stored at all. So whenever the stimulus occurs, once that happens, then the body starts producing your steroid hormones. So they're secreted about 10 to 15 minutes after the initial stimulation. Your peptide and protein hormones, they're not lipid soluble. Same with your catecholamines. But your dopamine, thyroid, and the rest of your steroid hormones, they're all lipid soluble. This is an important thing to remember because if they aren't lipid soluble, so your peptide, protein, and catecholamines, they can circulate freely in the blood. But once they reach their target cells, they activate cell surface receptors because they can't go into the cell. But all your lipid soluble hormones, so your other amines and your steroids, they need binding proteins to travel through the blood. But once they get to their target cell, they can go into the cell and they activate intracellular receptors. So this is just another summary slide. So we'll start by talking about the anatomy. So we have the hypothalamus, which is connected by the infundibulum or the pituitary stalk to both the lobes of the pituitary, the anterior and the posterior. The anterior lobe is also called the adenohypophysis, and the posterior is called the neurohypophysis. Those are good names to remember because sometimes they use them in exams instead of anterior and posterior pituitary. This section here, where the pituitary stalk joins the hypothalamus, that's called the median eminence. And that's where the capillaries recombine to form a portal venous system. So this system is called the hypothalamic hypophyseal portal vessels. Basically, the portal vessels between the hypothalamus and the pituitary. So that portal vessel system is very important because it allows any hormones from the hypothalamus to really quickly reach the anterior pituitary because they don't have far to travel. And it also means you only need a very small amount of hypothalamic hormones to have a big effect because there's such a small amount of blood in that area that the concentration of the hormones doesn't get diluted like it would in systemic circulation. So we have 6 hormones that go to the anterior pituitary and 2 that go to the posterior pituitary. So the hypothalamus basically creates these 6 hormones that go to the anterior pituitary to activate the pituitary to produce other hormones that go somewhere else. But for the posterior, it's a bit different. The posterior pituitary doesn't produce any of its own hormones. It's kind of just like a gateway for the hypothalamus. So the oxytocin and the ADH, they're produced in the hypothalamus and they're just secreted through the posterior pituitary. So we call them hypophysiotropic hormones. These are hypothalamic releasing or inhibiting hormones. So they're any of the hormones that the hypothalamus releases that controls the pituitary gland. So we have a 3-hormone sequence for these hormones. And this is what I'm just going to show you what a negative feedback loop looks like now. So you have some sort of stimulus. So like your blood pressure drops, you get cold, or you're stressed, and any kind of stimulus. And that activates your hypothalamus. The hypothalamus begins to increase secretion of a particular hormone. So the concentration of that hormone in the portal system increases, and that activates the anterior pituitary to increase secretion of a second hormone, and the concentration of that hormone in your systemic circulation increases, and then that reaches a target endocrine gland, so like your thyroid or your adrenal glands, and that then produces another hormone, which its concentration in the blood is increased, and that reaches its target cells wherever they may be. And those target cells then do whatever process they need to react to the stimulus. The negative feedback loop comes in where these red arrows show. So your third hormone from the endocrine gland, your target endocrine gland, that goes back. Sorry, it doesn't go back, but like when there's an increased concentration of that hormone in the bloodstream, then your hypothalamus and your anterior pituitary can detect that, and they stop producing as much of their hormones, because now they're like, oh, the concentration of this third hormone is so high that we don't need any more. That's enough to do whatever we're trying to do. So that's why they stop producing the hormones. So most systems in the body are negative feedback loops, because if they weren't, then we just end up with like an overflowing amount of the third hormone, and that would not be good, because instead of just correcting whatever the stimulus was, we would overcorrect it. There are a few exceptions. When we get to the posterior pituitary, we'll be going through some of the positive feedback loops. Okay, so we'll go through the hypothalamic and anterior pituitary hormones now. So the first one is GnRH, gonadotropin releasing hormone. The good thing about these is most of them you can kind of tell from the name what they're going to be, so you don't have to remember too much. So gonadotropin, you can tell it's probably going to go to the gonads. So first it goes to the anterior pituitary, where it increases the secretion of LH and FSH, which then go to the gonads. Don't worry too much about what LH and FSH do, because that will be covered in Repro next semester. Just remember that the hormones exist for now. The next hormone is GHRH, so growth hormone releasing hormone. So that increases the secretion of growth hormone. But there's also somatostatin. So somatostatin is an inhibiting hormone. So that decreases the secretion of growth hormone. And growth hormone just goes to the liver and other tissues. The next hypothalamic hormone is TRH, so thyrotropin releasing hormone. So you can kind of tell from the name thyrotropin that it goes to your thyroid eventually. So first it goes to anterior pituitary, increases the secretion of TSH, thyroid stimulating hormone, which then goes to the thyroid. Then next we have dopamine. So that's an inhibiting hormone. So that decreases that decreases the secretion of prolactin, and prolactin acts on the breasts. Once again, don't worry too much about that system. You'll learn about what prolactin does next semester in Repro. And your last hypothalamic hormone is CRH, or corticotropin releasing hormone. And that increases the secretion of ACTH, adrenocorticotropic hormone. And that goes to your adrenal glands. That naming system will make a bit more sense when we actually go through the adrenal glands, because when we do that, you'll find out that basically cortisol is one of the hormones produced by the adrenal glands, and that is the hormone that does the negative feedback loop. So then you can remember that that whole adrenal gland system is all the C hormones, then like your thyroid systems, all the T ones, and then your gonadotropin and growth hormone, it's in the names. So it's not too bad to remember if you break it down like that. Okay, so onto the posterior pituitary. So like I said before, the posterior pituitary doesn't make any of its own hormones. It's just an extension of the hypothalamus, basically, and just a gateway. So we have these neuron nuclei in the hypothalamus, your supraoptic and your paraventricular, and they make your two posterior pituitary hormones, which are vasopressin, which is also called ADH, antidiuretic hormone, and oxytocin, and they're both peptide hormones. So oxytocin has two major actions. It's involved in lactation and childbirth. Oxytocin is one of the exceptions to our rule, so it has two positive feedback loops. So we'll go through the lactation loop first. So basically the stimulus is when the baby is sucking on the nipple. And so that causes the hypothalamus to produce more oxytocin, which is then released via the posterior pituitary, which then stimulates the smooth muscle cells of the breast to allow milk let down. And basically, the hypothalamus keeps producing oxygen. Because there's no negative feedback loop, the amount of oxygen in the blood doesn't matter. The hypothalamus will keep producing, oxytocin. The hypothalamus will keep producing oxytocin till the initial stimulus stops. So once the infant has had enough, then the suckling stops, and then the hypothalamus will stop producing oxytocin, and milk letdown will stop. Similarly, for the childbirth loop, so this is basically during childbirth, when the baby's head is pushing on the cervix, the hypothalamus releases oxytocin via the posterior pituitary, which then goes to stimulate contraction of the smooth muscles in your uterus and the cervix. And that process keeps happening, more oxytocin is produced, there's more contractions until the initial stimulus is gone. So until the baby is born and there's no more pressure on the stretch receptors, more oxytocin will be produced. That's just another summary table you can look at later. Okay, onto vasopressin or ADH. So vasopressin is its main control is over blood pressure. So we have two triggers for it. So either an increase in osmolarity or a decrease in blood pressure. So it's very sensitive, our body is very sensitive to osmolarity. So an increase of just 1% can set this whole loop, can start this whole process. But it's a lot less sensitive to blood pressure. So you need a change of 10 to 15% at least for anything to happen. So an increase in osmolarity or a decrease in blood pressure will result in the hypothalamus releasing ADH via the posterior pituitary, and that acts on your V1 and V2 receptors. So your V1 receptors are in your blood vessels and it'll cause vasoconstriction. Your V2 receptors are in your kidney and they'll increase the reabsorption of water. So both those things together result in an increased blood volume and an increased blood pressure. So basically overall, increased osmolarity or a decrease in blood pressure contributes to secretion of ADH, which then results in an increased blood pressure. Just another summary slide. Okay, so onto the conditions. So we'll just start with growth hormone excess. So this is usually caused by a pituitary tumor, and it has two conditions that result from it, depending on whether you're a child or an adult. So if you're a child, you end up with gigantism, which is this first picture. So this is because your bones aren't fused yet, so they just lengthen, so you get really tall. But if you're an adult, your bones have already fused, so they just thicken and you get this thing called acromegaly. The only treatment for this is you can have surgery to remove the pituitary tumor, or you can use medications like dopamine agonists, somatostatin analogs, or growth hormone receptor antagonists. So basically, with the medicines, you're either trying to stop the secretion of growth hormone, or you're trying to make the body insensitive to the extra growth hormone. So if you can have excess, it makes sense that you can also have deficiency. So we have two types of deficiency of growth hormone, idiopathic and Laron dwarfism. So the idiopathic one, well, it's idiopathic, so it has no cause. So you just treat it by injecting growth hormone for as long as the child is growing. But for the dwarfism, the issue is you have normal GH levels, but your body isn't sensitive to it anymore. So giving them more growth hormone will not help, as the body will still be insensitive to it. So what you do is you give them IGF-1 as an injection. So IGF-1 just helps the growth hormone with its process. Okay, so diabetes insipidus. This is a deficiency of vasopressin or ADH. Insipidus means tasteless. So the reason it's called diabetes insipidus is because patients with DI produce a large volume of hypertonic, dilute, and tasteless urine. And that's usually accompanied by an increase. They become really thirsty and drink a lot of fluid. So there can be two causes for this. So either a central cause where they're not producing enough ADH, or a nephrogenic cause where the kidney is not sensitive to ADH anymore. So we can do a test to try and figure out which cause it is or if it is DI at all. And it's called the dehydration test. So basically you stop the patient intaking any fluids and you measure the urine osmolarity every hour or every so often. So if you look at this graph on the right, so at the bottom we have the number of hours after they last had water, and on the on the y-axis you got the osmolarity. And so normally we retain water, but we lose the solutes. So our osmolarity increases, which is this red line over here. So it increases pretty quickly and it just stays up here. But if we have DI, we can't retain the water. So we lose a lot more water with the solutes, and so our osmolarity doesn't rise that much. So they're all around at least half of normal. We also do this test called, so we use desmopressin. So desmopressin is a vasopressin analog, and so we give that to them. And so that can differentiate between a central cause and a nephrogenic cause, because in a central cause, they're not producing ADH. So giving desmopressin basically fills in that need. And so their body should sort of behave more normally after that. As you can see with this green and this dotted red line, they both increase rather dramatically after being given the desmopressin, which indicates that it's a central cause because the desmopressin aided them in being more normal. But if it's a nephrogenic cause, the kidney is being insensitive to any ADH. So giving desmopressin will not have any effect because the kidney will still be insensitive to the desmopressin. And that's why this blue line down the bottom, there is no change at all. And one last slide. So basically, if you're drinking a hypertonic solution, so like water, your plasma osmolarity will decrease. And therefore your ADH secretion decreases. And you'll end up with dilute urine. But if you're drinking a hypertonic solution, or you're exercising a lot so you're not intaking enough fluids, then your plasma osmolarity will increase, and your ADH secretion will also increase to help your body retain water, and you'll end up with really concentrated urine. And that's all I have for today. Remember, you can message us if you have any questions or use Curious Cats. Thank you.